One and all. Today I'm going to talk about my project on analysis of hepatocellular carcinoma transcriptomic data for drug repurposing. Let me first of all introduce myself. I'm Sonalika Ray. I've done my master's in bioinformatics from Punjab University, Chandigarh, and my research interest lies in omics data science, computer-aided drug designing, and pharmacoinformatics. So my topic is hepatocellular carcinoma. I'll be first of all shedding some light on what it exactly is. So it is the second leading cause of cancer-related mortality globally. It is present in patients with chronic liver inflammation associated with viral infection, alcohol overuse, or metabolic syndrome. A significant progress has been made in HCC prevention, diagnosis, and treatment in the past. It has been seen that more than 50% of all the HCC patients have a diagnosis at an advanced stage, that is a stage B or higher, and 70% of patients relapse within the first 5 years of initial treatment. Early HCC is often resectable, but advanced HCC often requires Sorafenib for systematic treatment in addition to local treatment with ablation, transatrial chemoembolization, or external irradiation. Sorafenib is effective in improving the outcomes of HCC patients in the late stage, initiating a period of robust clinical research. But it has been observed that Sorafenib gains resistance in the later stages. So let's see what the mechanism of action of Sorafenib drug is in HCC. Sorafenib is a multiple target tyrosine kinase inhibitor that exhibits anti-angiogenesis and anti-proliferation effects and extended total median survival in advanced HCC patients. It suppresses tumor cell proliferation by inhibiting RAF1, BRAF, and kinase activity in the RAS, RAF, MEK, ERK signaling pathways. It is capable of targeting the very three targets PDGFR beta, VEGFR2, c-KIT, and other proteins to inhibit tumor and angiogenesis. Lenvatinib, Regorafenib, Cabozantinib, and Ramucirumab have received approval as second-line treatments after Sorafenib drug. Immunotherapy for HCC has not yet been approved in China or Germany. Sorafenib remains a cornerstone treatment in HCC that is supported by robust evidence and clinical experience. So 30% of the patients they get benefit from Sorafenib, and this population usually acquires drug resistance within 6 months. And also a role of epigenetics, transport processes, regulated cell death, and the tumor microenvironment in the initiation and development of Sorafenib resistance in HCC has been seen. So what are those pathways that could lead to hepatocellular carcinoma and Sorafenib resistance hepatocellular carcinoma? So I'm going to abbreviate these two terms throughout my presentation as HCC and SRHCC respectively. So you see there could be a number of agents that could affect a number of pathways leading to HCC. It could be because of aflatoxin, alcohol, also chronic hepatitis B or C could affect the pathways leading to HCC. And to talk about the pathways that could get affected, which are responsible for causing Sorafenib resistance in HCC. So Sorafenib is an anti-cancer drug that is transported into the cell through SLC proteins. And inside the cell, the drug is broken down via various phases and cascade of reactions. Sorafenib is a drug that is designed to target multiple tyrosine kinase inhibitors such as VEGF, PDGF, and also EGF that activates downstream signaling pathways such as RAS, RAF, MEK, ERK pathway. Overexpressed drug targets and perpetually activated signaling pathways will promote cell proliferation and survival, therefore promoting chemoresistance. Cellular pathways or processes such as DNA repair, apoptosis, and autophagy can redirect their effect to promote cell proliferation and survival rather than cell death. I will also shed some light on the role of MAPK signaling pathway and PI3K AKT pathway in causing the condition. So now talking about the research plan, I took two data sets. They were from the cell lines HepG2 and HUH7, matched the gene expression alteration in resistant HCC with gene expression changes caused by treatment of cancer cells with drugs already FDA approved for other diseases, identified the drugs that could reverse the resistance related changes, and also then identified their mechanism of action, the pathways or the drug target involved of the drug against HCC. And finally, provide proof of concept evidence for validity of the drug repurposing approach with potential use in personalized medicine. So the datasets that were taken were two of them. In the first dataset, the samples were from a normal population and the HCC patients, and the second dataset had samples from HCC patients and Sorafenib resistant HCC patients. Now, the first data set that was taken, it was evaluated for the differentially expressed genes by carrying out the various steps required, such as a normalization and then the statistical tests, including the calculation of p-value, adjusted p-value, and log-fold change. And then the entries of the p-values, the gene IDs, and the log-fold change values were all together taken and submitted on iLinks for further evaluation. After the evaluation of my entries of differentially expressed genes and also the p-values and the log-fold change values, I found out some of my results that I'm going to share with you all. So this is the result for transcription factor enrichment analysis, which tells that SOX2 and HNF4A are the most differentially expressed transcription factors, and the kinase enrichment analysis result shows that MAPK1 and MAPK3 are again the most differentially expressed kinases, or we can also say that they are statistically significant. And why are they now differentially expressed is the question. And for the same data set, I also found out the protein-protein interaction expansion, where I found that ESR1 is that one protein which, when affected, would lead to a cascade of reactions to affect the other proteins. And then the expression to kinases network is also shown very beautifully here, which shows that when the ESR1 protein is stimulated, it's actually going to affect these various proteins and also the kinases that have been very well mentioned. Now, what are the hub genes for HCC? So, GPC3 was a particular gene that I found in the gene linear result that actually also affects a lot of other differentially expressed genes that were found in my dataset. So now, what is GPC3 gene all about? It positively regulates the canonical WNT signaling pathway by binding to the WNT receptor frizzled and stimulating the binding of the frizzled receptor to WNT ligands. Also, the result was validated from Signet A results that shows that AFP and GPC3, these two genes were the most differentially expressed genes and also are the main hub genes, right? After this, I took my results, that was the list of differentially expressed genes, the gene IDs, the p-values, and the log-fold change values to L1000 CDS square. Now, L1000 CDS square is a Lynx L1000 characteristic direction signature search engine. It enables users to find consensus L1000 small molecule signatures that match user input signatures. So my idea here was to reverse the effect of differentially expressed genes. Now, L1000 CDS square helps us with a list of perturbations and combinations that could be used effectively to reverse the effect of differentially expressed genes. Now, from the result of the very first analysis of the very first data set, I know what are my differentially expressed genes. proteins and kinases, though I just took the genes in consideration, but I would love to do my analysis with the differentially expressed proteins and kinases and EX2 and X2K expression network analysis as well. But yeah, coming back to the differentially expressed genes, I could find out a list of perturbations and combinations, which I further took to for further analysis on PubChem and also the DrugBank to see which are those perturbations which are FDA approved, first of all, and also who follow the Lipinski's rule of 5, right? The same particular gene set was taken further for analysis, and I found the genes. So here we have all the genes, and on top we have all the perturbations. Now these are those perturbations which are actually going to reverse the effect of these genes. So here in this particular graph, the input gene signature is depicted as the rows of the matrix, and the expression level of the genes are indicated with red or blue label bars indicating up or down expression. The top ranked L1000 perturbations are showed as columns with red label bars, and L1000 perturbation gene signatures are depicted as columns of the matrix with red and blue squares indicating their effect on the gene expression. So again, from here I could find out the list of all those perturbations which are actually reversing the effect of our differentially expressed genes. Now comes the second data set that had the samples of Sorafenib resistant HCC patients and HCC patients. The same thing that was done for the previous data set was done here as well. Differentially expressed genes were evaluated after running the normalization and calculating various p-values, adjusted p-values, log-fold change values, and then those values were taken to L1000 and I-lengths again. So this is just a screenshot that I wanted to share just to show how the signatures are submitted, and it's very easy to just upload and carry out the analysis. And here I again found the transcription factors that are differentially expressed that include TP63 and HNF4A, and kinases were the same. MAPK1 and MAPK3 again the differentially expressed kinases. And here SMART3 is that one protein which when affected would lead to the cascade of reactions to affect the other proteins as well, and is statistically significant. And the expression to kinases expansion also shows that SMART3 is that one protein that is affecting or interacting with a lot of differentially expressed proteins and kinases. The hub genes for Sorafenib resistant hepatocellular carcinoma was majorly MYOF. It is that one gene that interacts with other crucial differentially expressed genes, which leads to Sorafenib resistance activity in hepatocellular carcinoma. Now, again, the perturbations and combinations for the second data sets were evaluated, and also so in total I got 30 to 40 of perturbations and some 20 combinations, and I checked for each of the molecules activity on PubChem, on DrugBank, and yes, did check if they obey Lipinski's rule of 5 and are FDA approved or not. After all the analysis, this is the list of perturbations for normal versus HCC, and the perturbations for SRC versus HCC, and here SRC is actually Sorafenib resistant clone versus HCC, and the list of combinations. Now my idea here was to pick those perturbations that are actually common between normal and HCC dataset and Sorafenib resistant and HCC dataset, just to make sure that the molecule or the perturbation, let's say, that could be repurposed should be common for both the HCC patients and the SRHCC patients, and same for the combinations. So I further went to Veni and just looked for the common perturbations, and combinations. This is the result what I got. I got 10 perturbations that were actually common between both of the data sets. Now I studied the mechanism of action for each one of the perturbation that was common. So let's talk about the pathways in brief. The first is Selumitibib. It is an orally active small molecule with potential antineoplastic activity, and it inhibits MEK1 and 2 and prevents the activation of MEK1-2 dependent effector proteins and transcription factors, thereby leading to an inhibition of cellular proliferation in various cancers. Now, if you remember, I did say that we're going to talk about the affected pathways very soon in the presentation. So now let's talk about Afatinib. That is the other perturbation that was common in both the datasets. Afatinib is an orally bioavailable anilinoquinazoline derivative and inhibitor of the receptor tyrosine kinase epidermal growth factor receptor family with antineoplastic activity. The third common perturbation was Gefitinib. It is also an anilinoquinazoline with antineoplastic activity, and it inhibits the catalytic activity of numerous tyrosine kinases, including the epidermal growth factor receptor, which may result in inhibition of tyrosine kinase dependent tumor growth. Specifically, this agent competes with the binding of ATP to the tyrosine kinase domain of EGFR, thereby inhibiting receptor autophosphorylation and resulting in inhibition of signal transduction. Gefitinib may also induce cell cycle arrest and inhibit angiogenesis. The fourth common perturbation was Canertinib, that has a role as a tyrosine kinase inhibitor and is an antineoplastic agent. Lapatinib is a synthetic orally active quinazoline with potential antineoplastic properties. It reversibly blocks phosphorylation of the epidermal growth factor receptor and also inhibits cyclin D protein levels in human tumor cell lines and xenograft. The second last perturbation though was Vorinostat. It is a synthetic hydroxamic acid derivative with antineoplastic activity, and this agent also induces apoptosis and sensitizes tumor cells to cell death processes. Vorinostat crosses the blood-brain barrier. And the seventh perturbation common was Mirdamitinib. Now, why have I specifically written about it separately? The reason being, after getting all my results, I went through some of the publications just to learn more about these perturbations, and I got to know that these perturbations are already in the clinical trials to treat Sorafenib resistant HCC. So yes, I can call my project a case study, but my goal here was to repurpose a drug molecule. So Mirdamitinib was that one molecule or a perturbation about which I could not find more with respect to its being in clinical trial or so. So yes, let's learn about Mirdamitinib. Mirdamitinib is an orally bioavailable synthetic organic molecule that is targeting mitogen activated protein kinase with potential antineoplastic activity. And upon administration, Mirdamitinib selectively binds to and inhibits MEK, which may result in the inhibition of phosphorylation and activation of MAPK, ERK, and inhibition of tumor cell proliferation. The dual-specific threonine tyrosine kinase MEK is a key component of the RAS-RAF MEK-ERK signaling pathway that is frequently activated in human tumors. Now talking about the common combinations between both of the datasets. So the results that I obtained were Selumitibib plus DLPDM, MP and Mitoxantrone. The results of DLPDMP were not satisfactory when I took it to PubChem and DrugBank, and that's why I just picked Selumitibib and Mitoxantrone to talk about that could be used as an effective combination to treat sorafenib resistance in HCC patients. So the first perturbation in the combination is selumetinib that inhibits oncogenic downstream effects of the RAF-MEK-ERK signaling pathway, and these are the interacting gene partners that I've mentioned here on the slide. And then mitoxantrone is a potent inhibitor of topoisomerase 2, that is an enzyme that is responsible for uncoiling and repairing damaged DNA. And again, the interacting gene partners of mitoxantrone have been mentioned here. You can pause the slide and refer to them if you would want to. Now, what the conclusion of this project would be? The conclusion is that the drugs mentioned, Selumitibib, Gefitinib, Afatinib, Mitoxantrone, Mirdamitinib, Lapatinib, Canertinib, Trichostatin A and Vorinostat could be used to overcome Sorafenib drug resistance, as these drugs tend to affect the pathways leading to cell apoptosis. Now, what other differentially expressed genes that were found have been mentioned here? GPC3, AFP, LPS, RPS4Y1, MGLL, SERPING1, MYOF, MAP1B IGFBP4. Now, our study was centered towards repurposing drugs from differential activity of genes, taking in account the differential expression of genes and their role in the affected pathway. The drugs proposed for repurposing have been mentioned already. So we show the feasibility of the drug repurposing workflow by validating novel drugs to use alone and in combination with sorafenib in HCC. And the analysis here of the publicly available gene expression datasets of sorafenib resistance models and HCC patient datasets was done to determine prioritized drug candidates that may inform future additional validation studies. And I wish to study more about the role of differentially expressed transcription factors, differentially expressed kinases, and also look at the epigenetic effect that leads to sorafenib resistance in HCC. So that was it. I am looking forward to your feedback. Let me know in the chat box if you have any feedback for me. Thank you.